Hello everybody and welcome back to Creation Myths. Today we're going to start something called Creationist Contradictions, and we are going to talk about the waiting time problem and created heterozygosity. Before we get into it today, I have to explain what we're doing here. Basically, creationists have made a whole lot of sciencey sounding arguments, and they're just all wrong. They're all nonsense. I'm talking about things like genetic entropy, created heterozygosity, Dr. Nathaniel Jeanson's time to most recent common ancestor calculations for the mitochondrial DNA and the Y chromosome. It's all just nonsense top to bottom, but I've covered a lot of it already. I feel like I've covered most of it already in the main Creation Myths series. There's a few big things I'm still going to tackle. I have some stuff in the works, but I've covered a lot of the big stuff. So in addition to looking at individual creationist arguments kind of one at a time, I also want to look more holistically at the model that creation scientists are building. Because when you look at these arguments together as a whole and see how they work with or against each other, what you see is not only are these major creation science arguments wrong, they're actually contradictory. They actually invalidate each other when you dig into how they're supposed to work. So in this series, we're going to show how some of the big creation science arguments are actually incompatible with each other, and how creationists, either realizing this and not caring, or just not realizing it, make incompatible arguments at the same time as part of their models. The two arguments we're going to talk about today are the waiting time problem and created heterozygosity. I've talked about both of these before in kind of my regular debunking creationist arguments videos. Those are both going to be linked down below. So if you want the details on how these arguments are supposed to work and why they're wrong, check out those videos. But what we're going to do today is just very briefly reintroduce these two arguments and then look at how they're incompatible with each other. So we'll start with the waiting time problem. The idea with the waiting time problem is it takes too long to get new things through evolutionary processes. Basically, we're talking about like combinations of mutations or genetic traits. Okay, you don't have a trait and you have to evolve that trait, and it has so many steps and it would take too long to evolve through that pathway. That's the waiting time problem in a nutshell. It's an extremely popular creationist argument, and it's even something they've snuck into secular journals from time to time. So here we have John Sanford et al. Writing in the Theoretical Biology and Medical Modeling Journal. And then over here we have Ola Hofstra, Gunter Beckley, and Ann Gager in the Journal of Theoretical Biology. I have a lot to say about these two journals. For now, I'll just say they are creationists' favorite journals and leave it at that, but I should probably cover them at some point. Our second argument that we're going to look at today is created heterozygosity. This is creationists' attempt to answer the question, how do humans have so much genetic variation after just 6,000 years? If we started from two people 6,000 years ago, how do we get all the variation that we see today? Well, the answer, according to many creationists, is created heterozygosity. Adam and Eve were heterozygous, meaning for each site in their genome, they had 4 different versions of it, right? Adam had 2, so Adam was heterozygous, and Eve had 2, so Eve was heterozygous. And for a given gene, right, you could have 4 versions of that gene, 4 alleles, but they wouldn't just differ by 1 nucleotide. They would differ at a lot of nucleotides in those genes, and then through recombination, you could generate all the new variants we see today. So here is a paper in Answers Research Journal by Dr. Nathaniel Jeanson and Dr. Jason Lisle, looking at this created heterozygosity hypothesis. The specific version of this that Dr. Jeanson likes is called the CHNP model, the Created Heterozygosity in Natural Processes model. Again, the idea is Adam and Eve are created with lots of variation, and then you mix and match it to generate all the variants we see today. So what are the contradictions between these two arguments? Well, the waiting time problem presents evolution as a serial rather than a parallel process. What this means is just like this simple figure right here. You need to have mutations occurring, or changes, whatever the case is, evolutionary events occurring in a sequence with each other. Event number 1 happens, and then event number 2 happens, and 3 and 4, and however many you need in a given pathway. This is from that Hofstra et al. 2021 paper. They wrote, it is further assumed that the mutation rate is small enough to warrant a fixed state population, so that all individuals have the same regulatory array at any given time point until the next successful mutation arrives in some individual and spreads to the rest of population. What this means, if you simplify this language, what they're saying is mutation number 1 happens and then achieves fixation, so everybody in the population gets it, and then mutation number 2 can happen and achieve fixation, and then number 3, and so on down the line, but you have to do it in sequence. Now, created heterozygosity, on the other hand, correctly, I will say, treats evolution as a massively parallel process. This is from Dr. Nathaniel Jeanson's 2017 book, Replacing Darwin. He says, if new varieties appear each generation, if millions of DNA differences can arise in a single generation, then the isolation of genetic and visible distinctiveness is comparatively easy. No need to sequentially isolate or select individual DNA variants for generation after generation. No need to sequentially isolate or select individual DNA variants. He's saying because you can have lots of people within a population experiencing evolutionary changes, lots of members of a population experiencing evolutionary changes, you don't need to have mutation number 1, then number 2, then number 3, then number 4 in order. You can have lots of things happening all at once in different lineages within a population, followed by, as we'll see, recombination. So right away, these are not compatible with each other. It's one with a serial process or the other with a parallel process. But it gets worse, because the waiting time problem also requires that there is no recombination. There's no shuffling of the deck from generation to generation. Again, from that 2021 paper, the authors write, it is reasonable to then believe that recombination between two parental regulatory sequences can be ignored. Now, I covered this in more detail in my video specifically on this, so I'm not going to go into the details now in terms of why that's wrong. It doesn't matter. But let's just see what the proponents of created heterozygosity have to say about recombination. Here we go. Turns out, created heterozygosity not just involves, but requires recombination. A lot of recombination. A lot of extremely rapid recombination. So again, going to Jeanson and Replacing Darwin, he writes, mutations might increase the diversity slightly, but around 99% of the DNA differences between chromosomes would be due to inheritance, and those differences should be easily classifiable into one of the 4 starting chromosomal versions in the original parents. So here we have Jeanson saying 99% of the variation we see, it's not due to mutations, it's due to recombination from those 4 initial variants that were created. And then we can look at Sanford et al. Dr. Rob Carter is a co-author on this piece that I'm quoting here, and I forget, there's another co-author, but I forget who it is. But they write, since each one of the 88 original autosomes in Eden, and by the way, that's the chromosomes 1 through 22, they're excluding the sex chromosomes because they do recombine, but if it's XY, it's a smaller region, so they're not dealing with sex chromosomes here. Just an aside, that's why it's 88. Since each one of the original 88 original autosomes in Eden could have carried its own unique set of designed variants, normal chromosomal recombination and segregation could have generated a large number of genotypes in the second generation. In each succeeding generation, allelic diversity could increase due to newly arising mutations and further recombination. Now, I want to highlight one very specific thing here. A large number of genotypes in the second generation. This is requiring not just recombination, crossing over between chromosomes, but crossing over at a rate that greatly exceeds what we observe today. In general, this is a rough estimate, but in general, you get approximately 1 crossover per chromosome per generation, or per meiosis is a more precise way to say it. That's not that many crossovers in general. You're not going to generate in a single generation that many new variants that way. If you can generate a large number of genotypes in the second generation, then you're not just leaning on recombination. You're invoking recombination at a speed that greatly exceeds the rate at which it occurs in the present. Again, I've talked about this in detail in the video specifically on created heterozygosity. I pointed out here to highlight the contradiction. We're not just talking about a little bit of recombination versus a little bit of more recombination. We're talking about no recombination is permitted for the waiting time problem, whereas for created heterozygosity, faster recombination than we observe is required. Warp speed recombination is what's required. So put these together, the serial versus parallel problem and the recombination problem here. And these two arguments are clearly contradictory. Creationists can make an argument for the waiting time problem, or they can make an argument for created heterozygosity, but you can't reasonably hold both positions at the same time. Or can you? Let's ask Dr. Rob Carter if you can hold both positions at the same time. Dr. Rob Carter is a marine biologist and geneticist with CMI, and here is a video from his YouTube channel. The waiting time problem is a huge problem for evolution. That's from his channel, Biblical Genetics. And so he obviously argues for the waiting time problem. I've actually covered this video, so you can find that linked down below. But he's a co-author with Sanford on that paper from the Proceedings of the International Conference on Creationism, so from 2018. So strictly speaking, it's not a paper, it's the proceedings from a conference, but technical distinction. So it was Sanford and Carter and somebody else on here. I forget the third author, and I cut it off to fit it on the slide, so my bad. They, at this conference, argued for created heterozygosity. That's kind of weird, isn't it? So you've got Rob Carter promoting the waiting time problem and Rob Carter promoting created heterozygosity, either not realizing that they're mutually contradictory or not caring. To wrap up this first installment in creationist contradictions, there are a lot of sciencey creationist arguments out there, but many of them are incompatible with each other. That doesn't stop creationists from using them anyway. Case in point that we just looked at is the waiting time problem and created heterozygosity. Ignoring that both are wrong, they're basically exact opposites in terms of how they treat evolutionary processes, right? The waiting time problem treats evolution as a very linear process involving no recombination, and created heterozygosity treats evolution as a massively parallel process involving lots of recombination. They are completely incompatible with each other. So I want to end this first installment in this series with a request and really a plea to creationists. Please, when you are constructing a model and you have arguments, look into those arguments, make sure they are consistent with each other. And when you find that some of the arguments you're making are mutually inconsistent, they are incompatible with each other, please pick one. Don't just use both and hope that nobody notices, because guess what? We're going to notice. So thank you all for watching. I hope you enjoyed this first installment in what will be an ongoing series. If you enjoyed this, hit like. If you did enjoy this, hit dislike. Either way, hit share. And promote it to people as good science communication, or promote it to your creationist friends as an example of somebody who really gets on your nerves. It's all the same to me. Please click one of the buttons down below. And if you haven't subscribed, please do subscribe. And as always, don't get fooled.